In this video, we're going to talk about the process of transcription. The overall process in making proteins is protein synthesis, and that's going from DNA to RNA. But then that's just copying the information or taking the information out to the ribosomes. And from there, the RNA has to go and make proteins from there. We're not going to get into all of that. We're just going to go through the first half of that and deal with transcription. So in other words, today for transcription, we're going to talk about going from a DNA nucleic language to an RNA nucleic language. We're just taking the information from one form of information to another form of information. The reason that we have to do this is because DNA is too large to leave the nucleus. The DNA is what holds all the information to make all of the different proteins. Now, we don't need all of the proteins on all of the time. We just need to make a particular protein at different times. So we'll have a signal that will allow for a gene to be turned on, if you will, and the mRNA for that gene is made at a particular time. So the mRNA is a smaller molecule, so we'll take the information from DNA in the nucleus and make mRNA or messenger RNA, and that messenger RNA can leave the nucleus. So again, transcription is going from the DNA language to the RNA language. So RNA, remember, is a ribose sugar rather than deoxyribose. It's single-stranded rather than double-stranded. And the nitrogen bases, RNA has uracil, not thymine. Still has adenine, guanine, and cytosine. The bonding rules, again, going from DNA to RNA. If it's G in the DNA, it'll be C in the RNA. If it's C in the DNA, match up with a G in RNA. If we see a T on the DNA side, we'll see an A on the RNA side. And if we see an A on the DNA side, it'll bond to a U on the RNA side. So transcription. It's a process in which an RNA strand is made or synthesized from a DNA template strand. So we're not making RNA from both strands. Remember that DNA is double-stranded. Well, when we go in and make our mRNA strand, we're only going to make mRNA off one strand. Okay, we're not going to make mRNA from both strands, just one of the strands. RNA polymerase is the enzyme that performs transcription. In DNA replication, there was lots of different enzymes involved. In transcription, for the most part, the biggest thing was to make the mRNA strand, we only have one real enzyme involved. And all of this transcription process happens in the nucleus. So why does transcription happen in the nucleus? Well, remember we said that DNA is a large molecule and DNA is housed in the nucleus. So, therefore, if DNA is too big to leave the nucleus, this process of getting the information or turning that code into an RNA language has to happen in the nucleus. So if all of this was in the nucleus, the blue strand here would be our two DNA strands. Our DNA strands are pulled apart, and our mRNA strand is made from our DNA template strand. Remember, just one of the DNA strands is used to make our mRNA strand. So again, like we mentioned before, the matching of the bases on how we're going to get a G on the mRNA strand is going to be matched up to a C. RNA polymerase is the enzyme that does this. If we have a T on the template strand in the DNA, an A will match up to it. If we have an A on the DNA template strand, RNA polymerase will bring a nucleotide of RNA with a uracil to match up. C's with G's, T on the DNA, match up, match up. So what should match up here with this A on the DNA template strand? Well, RNA polymerase will go in and grab a nucleotide with a uracil to match up here. with the A on the DNA template strand. RNA polymerase will then, to match up with this T, grab an RNA nucleotide with an A to match up. And then C, well, RNA polymerase will grab a G for that one, so on and so forth, until the entire mRNA strand is made. The big nitty-gritty stuff, okay? RNA polymerase is going to break the hydrogen bonds on the DNA strands. So the DNA strands go from connected together with hydrogen bonds between them, will go to spread out apart and break those hydrogen bonds to spread out the two DNA strands. RNA polymerase, the same enzyme, will bring in free-floating nucleotides to make the mRNA strand. Just like in the last slide where we showed you, RNA polymerase will bring in free-floating nucleotides of RNA to make an mRNA strand. And again, remember that's just on the DNA template strand. After that happens, okay, the mRNA strand will leave the nucleus by itself. When the mRNA strand leaves the nucleus, then the two DNA strands will come back together as though nothing happened. Eukaryotic genes, they have some junk, and we don't really mean junk, we just mean that there's sequences that are coding that are going to make the protein and some stuff in the middle that aren't for that specific protein. So exons are the portions of the DNA that will be expressed, or in other words, they're the good stuff. They actually code for our particular protein. The introns are the DNA sequences of DNA in between the exons. So down here in this graphic down here, in this blue color, we have the portions of the DNA or those spots in the DNA that will code together to actually make a protein. Where the stuff in orange, those are the introns, those are the things in between that go on that don't code for that specific protein. We have to get rid of the introns by the time this mRNA leaves the nucleus. So there's a little bit of work left to do, and the scientific name for this is post-transcriptional processing. So post meaning after, and transcriptional meaning after transcription. So the first thing that was happening was our transcript was made, our mRNA strand was made. So our primary transcript, or our pre-RNA, that's exons and our introns. So both of them together. Exons and our introns together. That's our primary transcript, or our pre-mRNA. But remember, we got to get rid of those introns. So we have to have mRNA splicing. These special things called spliceosomes are going to come through, and they recognize specific DNA sequences or sequences in the bases of the nucleotides. The spliceosomes, based on those sequences, will cut out the introns. So what's left behind is only the exons. So what we're left down here is called a mature mRNA transcript. So all we're left with is the exons. And that is what's going to go out to the ribosomes to be able to make the protein. So the protein, the mRNA strand that goes out to the ribosome only has exons in it to make the specific protein that we need. And again, that's called the mature mRNA transcript. So to recap part of the central dogma of molecular genetics, the overall big process of making a protein includes two parts. We have to go from DNA to RNA, and from RNA to a protein. We didn't talk about the whole process of protein synthesis, we just talked about transcription. And in transcription, we have our double-stranded DNA that's inside of the nucleus that is a part of a cell. what we're going to have is RNA polymerase is going to break the hydrogen bonds. Okay, when those hydrogen bonds are broken, the DNA strands separate and we make an mRNA strand off of one of the DNA template strands. And again, all of this is happening inside of the nucleus, that is inside of a cell. Then the mRNA strand is going to leave the nucleus. The DNA strands go back together. Again, that's in the nucleus. That's all a part of the cell. There's some post-transcriptional factors in there. We have to cut out the introns. And this mRNA strand is now going to look for a ribosome to go through and actually make the protein. We'll talk about that in the next video. And that is the transcription portion of protein synthesis, a small piece of the central dogma of molecular genetics. And that is all she wrote.
It's like this and like that and like this and it's like that and like this and like that and it's like this. So just chill till the next episode.